Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good afternoon, everybody, and welcome to the Florida Center for Individualized Medicine Grand Rounds. And as you all know, the SIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation and demonstrate how individualized medicine is being translated into practice to meet current and future patient needs. So then to that effect, it's my absolute pleasure to introduce today's presenter, Dr. James Cerhan. A little bit of background. Dr. Cerhan is the Ralph S. and Beverly Caulkins Professor of Cancer Research and Professor of Epidemiology in the Mayo Clinic College of Medicine and Science. He also co-leads the Genetic Epidemiology and Risk Assessment Program in the Mayo Clinic Comprehensive Cancer Center, and he also co-directs the Biorepositories program in the Mayo Clinic Center for Individualized Medicine. So Dr. Cerhan's efforts involve building and sustaining infrastructure to facilitate human research. And as just 2 quick examples of that, Dr. Cerhan has overseen the building of a biobank of 50,000 plus participants, as well as the collection of systematic data on all newly diagnosed cancer patients at Mayo, which is then used for state, national reporting, quality improvement, things like that, and of course, research to improve cancer care. So throughout Dr. Cerhan's talk, please use the Q&A function in Zoom to submit your questions. We have planned time for Q&A at the end of the session, and that's the main vehicle that we'll use. Now, for those of you who have not yet claimed continuing education credit, the code is Biobank. Biobank, one word, and we'll send out a message in the chat shortly to that effect. So without further ado, please join me in welcoming Dr. Cerhan as he presents on the Mayo Clinic Biobank, a precision medicine resource.
All right, thanks. Well, thanks for this opportunity. These are my disclosures, but none of them will relate to any of the content I discussed today. So we have 4 learning objectives: summarize the study design of the Mayo Clinic Biobank, our governance, describe our governance model, give examples of potential research applications. So I'll give examples that have been used, and hopefully it'll stimulate ideas for your own research, and then state how you can get information and actually propose a project. This is an institutional resource. This is the outline of the talk that I'll use as a guidepost so you know where we are, and it'll cover sort of where this came from, what's there now, and where it's going. So this goes back quite a ways to a task force led by David Showalter and Steve Thibodeau, that was set up by the research committee. And really they came out and wanted to know how to deal with biospecimens. And the key findings were that we really needed to maintain and build on some of the current institutional strengths. This was back in 2006, particularly around laboratory medicine, pathology, and epidemiology, but also really help leverage our existing disease-oriented collections that were getting large, but we're having trouble storing all their samples. But one of the things that came out as we interviewed people was the opportunity for what was then called a control collection. So the recommendations out of this task force was to establish oversight, expand the current infrastructure. So this really led to the investment in the BAP lab, RLIMS, pathology research core that we all know today, and then the establishment of the institutional collection of clinic-based controls, or what became known as the Mayo Clinic Biobank. So what is that? The initial goal was to recruit, consent, and obtain a blood sample and risk factor data from 20,000 participants, regardless of their health history. This would then be a resource for a wide variety of studies available to all Mayo investigators, and actually built in to have external collaborations, and then ultimately develop in a cohort, and if it was successful, scale it up to a larger group. Before we got going, so this is back when genetics was still fairly new, and so we did, through the work of Barbara Koenig, a bioethicist, a deliberative democracy. And this was really to get community input on what should this look like. And they did this, they selected a representative sample of 25 people across Olmsted County. They brought them in for 4 full days and gave them background into biobanking and genetics, and then really had them deliberate on, okay, how should we do this? And what they came back were some important recommendations. One, they said for recruitment, make it convenient, be clear in the risks and benefits. For informed consent, keep it simple, i.e. keep the lawyers away from the consent form. We were only moderately successful with that. State if there's going to be future contact, and ensure privacy, and articulate how that's going to be protected. They recommended keeping an ongoing communication with the donors in terms of both what the study's doing, and also if there were the potential for research findings to come back, people should have the option to receive those. And then for use, they really wanted internal and external use. They thought to maximize use, but they wanted oversight by Mayo Clinic and community engagement through the development of a community advisory board. And we took all those recommendations and actually implemented them. So the Biobank was actually launched 24 years ago this month, April 1st. It's a prospective cohort study. It's observational. There is no intervention. And it had very wide eligibility. Age 18 and older, you needed to have a Mayo Clinic ID. Living in the US, that's so that we could link to national registries, and ability to provide informed consent. We started in Rochester, expanded to Jacksonville in 2012, and then to primary care clinics in La Crosse, Wisconsin in 2013. Our consent form, this had been developing at Mayo. We really got this solidified. We worked closely with the IRB, because the consent form is what we call, and now Mayo uses widely, the registry consent, because we couldn't put in the consent form up front every study we were going to do. We couldn't have anticipated all of them, and we've had some very creative investigators. So this is really a more of a consent of a broad use of specimens and data in multiple research studies. I note research here. This results do not go into the clinical record, and I'll come back to that point. Access to past and future medical record data, use of clinical samples. So, for example, if you developed a cancer and you have tissue in the tissue registry, you'd allow access to that, as well as future contact, but only through the Mayo Clinic Biobank. And this could be through return of results or additional research. And we wouldn't contact anybody more than twice a year. And we actually maintain a way to track that, because as you'll see, we have gone back to these participants for special studies. We also received a certificate of confidentiality because we knew there'd be genetic studies from NIH. There was an incentive of a gift for those of you who may be on the call that are actually in the biobank. You probably chose the fleece blanket. Those are the most popular, or the water bottle. The postage stamps didn't get that big of an uptake. We did a baseline survey to get sort of these key areas, particularly these areas that are maybe not as well covered in the medical record. Biologic sample collection, we collected these through the clinical labs, for example, the Hilton Draw Center, or in Florida, places where you would send people for clinical draws. And then these were then transferred directly to the BAP lab, processed and stored. We tried to piggyback on a clinical draw when we could. That is, that after they got the clinical sample, we added on the research samples. This was important because the samples taken can be used for CLIA grade testing. And I'll talk about that in one of the projects where we sorted this out. Did a standardized collection, really to bank plasma serum DNA. We extracted DNA up front and then put away buffy coat as well. On 10% of the subjects, we did a heparin tube slow freeze, which has allowed us to bring back viable cells. We'll talk about a project that uses that. And then these were all aliquoted into robotic freezers. We were allowed access to the medical record data. Most of it, and you'll see this, is in electronic format, but we do have ability to go back to paper records if we need to. We'll talk about some of the data we extract out routinely from that. So here's sort of a schematic of the study. So at baseline, it took us from 2009 to 2016 to complete the enrollment. We did 20,000 first and then increased it to 50,000. And then we started a follow-up survey 4 years after enrollment. So you can see these, that started midstream when the first people enrolled in 2009 hit 4 years. We ran that out through 2019, and then we've done a second survey, but this time we did it cross-sectional. So it was just, we went to everybody in the cohort at the same time. And those were really to update exposures that, again, may not always show up so well in the electronic medical record. Well, who is in the biobank? First, I need to give credit to Janet Olson, who was really the co-PI with me on this and really led the recruitment. This is sort of a little summary of that paper. So we invited over 260,000 participants from appointment calendars. We had about 3,700 volunteers. Overall response rate of 21%, or about 56,000. That was actually better. We thought we'd get a 10% response rate. 76% of the participants when they were enrolled were from primary care clinics in Florida, Rochester, and La Crosse. And then 20% were from specialty clinics in Rochester, and then 5% were actually volunteers. We did some recruitment out at fairs and that with a mobile van. So here's a summary of the 56,800 or so participants. Skew female, that's who comes into for appointments, so no surprise there. And here you can see the age distribution with the peak between 61 and 70 years, suggesting this could be an excellent control group for chronic diseases that have a later age at onset. But we do know we do have over 6,000 participants under the age of 40 when they were enrolled. Where did they come from? The participants from the upper Midwest, 26% are from Olmsted County, which is in the blue circle there. Another 26% are from the state of Minnesota. Again, the darker colors are where counties where there are more participants from. About 14% from Wisconsin and Iowa, and 2% from the Dakotas. In Florida, 18% of the biobank are from Florida, and you can see the counties here that Jacksonville is in the dark color right there, and then particularly along the coast and from southern Florida enrolled. And then about 15% come outside of those regions from every state in the US and Puerto Rico. We did a little comparison to see, well, okay, we know this is a biobank sample coming in. They're going to be a little healthier because they're coming in for general medical exams, but how do they compare with population-based data? So here we compared the 10,000 enrolled in from Florida with the behavioral risk factor surveillance system. This is what CDC does to get population level data for Florida and Georgia. And you can see that we skew a little older enrollment. That's who comes in for healthcare. Not too bad on female gender, a little lower than the population-based estimate from the BRFS. But definitely more white and less Hispanic ethnicity and higher educated. We did the same thing here for first row here is the Mayo Clinic Health System, then here's the Rochester, and the same patterns exist. Older, more female, more white, less Hispanic, and higher education. We also compared a few variables. Here's for Florida. It looked pretty similar actually for body mass index, which is a measure of obesity, but less current smoking and a little bit more alcohol use gradient and fewer never users in the Biobank. And we saw that same patterns for the primary care clinics in La Crosse as well as the Rochester. In terms of follow-up, we do two types. The first is active, and this is via follow-up survey. And I showed you that on the outline there for the 4-year survey. So these are data here now based on survey. Here's at enrollment, so just the baseline survey. You can see the prevalence of 20 of the most common conditions. So you can see there's common things happen commonly, and we have lots of participants with them. And then for the subset of about 28,000 that we had both a baseline and a follow-up, they didn't die or get lost to follow-up, which there were some in here. You can see their incidence. So these are new onset conditions. For example, hyperlipidemia at baseline, they reported no hyperlipidemia. By 4-year follow-up, they had reported it. Now, this isn't perfect because, again, we lost about 22% through non-response. But we also follow people actively through the electronic health record. We defined a group here that we excluded about 15,000 of the 56,000 who really didn't have a very deep electronic health record. They, many of these probably came to the Rochester campus, had their hip replaced. We don't see them again. But for 40,000, we had actually very deep electronic medical records. So up here, you can see in 2009, we opened enrollment, and this is the enrollment of the participants. And then here you can see from the EHR, ICD codes, CPT codes, EKGs, et cetera, the number that we have, so many. And then this line here and all these shows the proportion of participants who had one of those in the given year down here. So you can see we have very deep EHR record data on over 40,000 participants, going back to really the dawn of electronic health records at Mayo, and then it continues to go forward, and we are able to electronically link in each year. And this just gives you some of the prevalent and incident numbers for that 40,000 for the top 20 US Department of DHHS chronic conditions. And as you can see, now you can start thinking of studies you might want to develop out of that based on your favorite phenotype. So part of setting this up, again, is, we couldn't tell people exactly what we're going to do in terms of the exact research. So we had to set up a governance, and then how do you access it? So in terms of governance, we set up the what's called BTOG, Biospecimen Trust Oversight Group, with representation from across Mayo, and really really set up a governance model. That is, it's going to have to evolve over time and address questions as they come up, as people want to use the biobank and new opportunities come up. That is linked carefully with the infrastructure. This has been key. This is the BAP lab and Pathology Research Core, because it's the underlying infrastructure for doing projects, and then closely linked with the bioethics program. And we'll talk a little bit about their role in this. So as a governance structure, we needed to keep participants informed. So there is an external website, and good news, you can just Google Mayo Clinic Biobank, and you'll come right to it. And we had a place where we keep information for participants. We'll talk about other uses of that. And then we do a regular newsletter to the participants called Bio News, where we kind of give them an update on how enrollment was going. We brought up the launch of the Community Advisory Board. Again, that came out of the community engagement, that we should have that and how it works. We give examples of studies that are approved for use. We'll talk about how that happens. We kind of give them a little background of, where is your blood going? How do we decide on access to it, and what are those principles? What's the Community Advisory Board regularly gives updates on what they're working on. We tell them when, like, for example, when we opened in Jacksonville, when we brought in new technology, like the bigger robotic freezers. And then as new technology came up, like when we were trying to move into whole exome and whole genome sequencing, just what is that? What are we thinking about it? And then our broader business plan. So lots of topics that really are viewed as a governance model, keeping people informed. Second component of it is our Community Advisory Board, and it really came out of that community engagement. This is selected because it's a rotating membership to broadly represent the community. We started in Rochester, we opened one and maintain one in Jacksonville as well. And they really provide advice and guidance on the management and operations, policies, when we want feedback on participant materials, like the follow-up surveys, and then complex decisions that come up. They've been a great resource to go to and get input. We'll talk about, so we have one for Rochester and one in Florida. We'll talk about the Sangre por Salud in a moment when we get to some of our sister biobanks. And this just gives you a wide range of what the CAB has given us input on: studies, return of results, the business plan, a whole variety of things. And they're available now as well. So you may be thinking of a study you want to do in the biobank, but you'd really like CAB input. There's a way to get that on a variety of these types of topics. They meet 4 to 6 times annually, and is coordinated by Karen Mahar. And for internal people, you can go to the Biorepositories program under community, and it tells you how to engage with them, or you can just send an email here. Third piece of the governance is the access, and we put an access committee that reports to the BTOG, really maintains the principles of use, reviews and approves projects, gives a prioritization, brings in expertise as needed, and again, we bring in very broad representation across multiple disciplines, and including a member of the Community Advisory Board. Our principles of use are probably what you would think they would be. They all have to have an IRB approval, in part to ensure both patient safety, but also to ensure that, okay, you signed a global registry consent. Does this broadly fit in, or is it different enough that you really need to go back and reconsent? It's reviewed by other scientists to ensure high quality science. We need a Mayo co-investigator because it needs to have an IRB, and that IRB has to be from the Mayo IRB for non-Mayo proposals, but we have a mechanism to bring outside researchers in. We do not allow researchers to identify the person except in a specific situation where we do a handoff. I'll talk about that. And then samples are only given for a specific purpose. And if you want to do more with it, you need to come back and get approval for that. But we do allow what we call super users. So if you set up a case control study and you know what you want to do for the first couple of years, but you'd like to make sure you have samples available, we have a mechanism to do that. Prioritization is really around science and patient interest, and then other priorities within the Center for Individualized Medicine. So we've improved to date 416 projects. We've only disapproved 2. Sounds like a committee that's pretty lenient, but basically a lot of this gets weeded out through the feasibility process, and I'll talk about that later, such that by the time it actually comes up for review, it's been reasonably well vetted. Well, what about the uses? How can I use this? So there's a variety of studies, designs. I'll just kind of highlight an example of each of these because I think it stimulates possibilities. First is just a very descriptive or prevalence survey. So here's an example that Yuijing Ru did with an early cut of the enrollment at a little over 21,000 participants, and asked the question, well, what is the prevalence in that sample of a self-report of a fair or poor self-perceived health, which turned out to be about 9.5%. And then what percent had 3 or more depressive symptoms on the PHQ-2? That was almost 5%. You could see here for general health that fair or poor health increased with age for men, but was fairly steady for women. So for most of these, it was higher in women until you got to the oldest age group here. While in contrast, depressive symptoms decreased with age and tended to be higher in women, although it varied a little bit, still fairly close. She also did an analysis then that said, well, most of the drivers of that prevalence rate was really due to disease burden, a bit to age, particularly for depressive symptoms, because you do see this strong gradient there, and then more weakly to these other factors like employment, alcohol use, education, body mass, smoking. Greg Pollan was interested in herd immunity around rubella virus. So he selected a sample from Olmsted County. The serum samples were banked, so they were just pulled. He could select this for people aged 20 to 44 and did a sero prevalence survey on that. Found that there was pretty good immunity, only negative in about 2%. But interestingly, about there's a pretty large sample under antibody titers under 25, which could be a little bit worrisome in terms of either they're having waning immunity or they never really responded. So it sort of gave a population perspective. Correlated that, really saw no correlation with titer levels and age or body mass index. Second use is as a control group for a case series, or essentially a case control study. And that, that was the original rationale to launch the Biobank, and has been actually one of our most common, probably the most common use. 2 examples. One, here's a genetics study led by Susan Slager, looking at a polygenic risk score. So in a large consortium, and this is pretty common with genetic studies that need very large groups, they had found a nice discrimination between controls here and cases here in an independent international study, and then came back and looked in her own familial cases, controls, and then here's the Mayo Clinic Biobank controls genotyped. So nice control group, actually aligned really nice with the international control group, suggesting, we're, at least for these genetic markers, we don't have something that's very odd. And really showed nicely how a polygenic risk score in this setting, CLL, chronic lymphocytic leukemia, has a very high familiarity, but you could actually find almost independent discrimination of risk of CLL independent of family history. That is, this polygenic risk score told you something in people without a family history and actually gave you even additional information in people with a family history. They also extended this to a precursor of CLL, showing the similar association. Here's a multi-omic study using plasma samples led out of Akhilesh Pandey's lab. Here he had 455 COVID patients on 3 levels of severity. So outpatient patients with severe COVID, patients with critical COVID, so went into the ICU. And he needed controls. He selected 182 age and sex match controls. SIM actually put this biobank together, so similar approaches were used for collecting the samples. So this was a very reasonable control group. He profiled a large number of circulating cytokines, proteins, lipids, and metabolites, and then using a machine learning-based prediction model, identified this subset of biomarkers which predicted severe clinical COVID-19 better than just sort of traditional cytokines. So this is an example of using the banked plasma samples. next is, so you can now do, for some common things, you can do a case control study completely embedded in the biobank. And just one example here, this is a little specialized, but on our first survey, we got the Rome-3 criterion for irritable bowel syndrome included. And so they got over 40,000 responses back. We're able to pull out from that 4,000 IBS cases, and then went into the remainder and did an age-sex matched 3-to-1 control, so about 12,000 controls. And then they did a latent class analysis, identified 7 distinct clusters shown here, demonstrating varied degrees of gastrointestinal symptoms, comorbidities, dietary and lifestyle factors. And one of the goals here that they're trying to do is see if they can maybe parse subtypes within here that could then more individualize clinical trials and patient management. Okay, this is a cohort study ultimately, because we're enrolled people at baseline and we're following them forward in time. 2 examples here. This is a Paul Takahashi's work where they took the biobank participants who are in PCIM, primary care internal medicine in Rochester, and with the houses geocoding developed by Young Jun, classified all the participants on a tier houses quartiles. So, and this is really a SES, social determinants of health kind of measure, and then cross-correlated that with Minnesota medical tiering. So think of it as a comorbidity index, and saw a correlation that as you had lower SES, you also tended to have more multimorbidity. But even when you put that together, being at the lowest quartile of, or the first quartile, which is shown here, the lowest quartile or lowest socioeconomic status on this measure, you had an increased risk of hospitalization, about 50% higher than those in the 4th quartile. And this was independent of age, sex, and the number of comorbidities you have. So it was giving you some additional information on risk of hospitalization. Susan Slager used the, so I recall we put a slow freeze down. She was on about 10% of the samples. She took those samples that were from the sort of Olmsted local region, and you could bring those back and do flow on them to identify the prevalence of the CLL precursor called B-cell lymphocytosis, and was able to show that and this was restricted to people 40 and older, that 17% had this precursor condition to CLL. Most of it was what's called low count, or would be completely missed clinically. Showed a strong association, the prevalence of MBL with age, and higher in men than women, which would parallel what we see for the epidemiology of chronic lymphocytic leukemia. But then was able to follow them forward in time and show that the MBL cases had about an almost 8-fold increase of lymphoid malignancy, and that existed both for the high count, which is sort of the subclinical CLL, but also almost a 4-fold increase for low count. So this is fairly common in the population. So shows how you can do a cohort, and that now has really led to investigating, well, can we predict who in the population should we be worried about with low count MBL that is at the highest risk of going on to developing a lymphoid malignancy. This makes itself very available to EHR-based research studies. And here's just an example linking this with the Rochester Epidemiology Project. That's about 14,000 participants. Vanessa Kronzer and Cindy Croson identified RA cases, did a manual review, and then applied a eMERGE as a large consortium nationally algorithm, and showed the performance of that, and showed that it improved with longer RA duration as well as a longer EHR duration up to 10 years, and then sort of the AUC maxed out, which then gives you some good insight on how to do case finding. Genotyping driven research is another approach. So in this study, Adrian Vela wanted to test the hypothesis that this genotype in the TCFL2 might impair insulin secretion. So he randomly selected out of the Mayo Clinic Biobank participants aged 20 to 70, no history of diabetes, lived within 100 miles of Rochester. So you can do that from the sampling frame. He genotyped all of those, and then he found this isn't a very common variant, but he found the variant 60 participants with it. From that genotyping, brought in 60 of the sort of normal genotype, and brought him in for additional testing, and then was able to show that TT genetic variant impaired glucose tolerance through effects on glucagon as well as insulin secretion. So it's a way to sampling frame, identify people. And this is where, because we can recontact people, we can bring them in. Finally, a couple large initiatives just to put on your radar. The first is the RIGHT protocol. It's probably been presented here several times: right drug, right dose, right time. This is really a preemptive pharmacogenomic testing, one of the real large successes of SIM. And the goal here is really just to, if we had the genotype in the medical record at the time somebody's being prescribed a drug, we would like to alert for the potential bad outcome. I think of a different drug. The first phase recruited about 1,000 biobank participants, sequenced 86 of them, returned that information to the medical record. Then it was expanded, and this work was also led by Li Wei Wang and Dick Wenchelbaum, who I'm sure you're all familiar with, for an additional 10,000 in the phase 2. And here you can see the high-level results for the genes from table 1. And the interesting thing here is that almost 80% of those over 10,000 participants had actionable variant in 3 or more genes, so very clinically relevant. The Biobank really facilitated this. We talked about, you can find subjects for your study. In the pilot phase, there was a much more extensive criteria to get people in. They found that didn't work all that well, and for phase 2, they just focused on non-white, so they wanted to enrich for diversity, and then EHR length. It's an efficient way to do recruitment. You can get really decent participation rates. For the pilot phase, they had to draw a second blood sample. But for the phase 2, we got the CLIA acknowledgement so that they were actually able to use the bank DNA to pull out and send a Baylor for the CLIA test, which really saved a lot of resources. And again, they were able to bring in existing survey data. And again, they needed to go back to participants. And here's where you do have to do a new consent, because the original consent said we weren't going to put anything in your medical record. So we had to consent that people were willing to do that. We ensure reuse of data. These data are available internally through the Center for Individualized Medicine. And there's a website. On the website, you can find out all the details and how to apply to use it. And you can actually, for external users, go to the SIM website under the pharmacogenomics program, find out more and how to collaborate. There it is. So through the 2010s, we were thinking about, boy, wouldn't it be nice to have everybody genotyped? This is where we stood. We had some GWAS data, not much sequencing data. So we were looking for ways to do that, both with our own resources and through partnering with companies in particular. And really, we wanted to expand to both whole exome sequencing and SNP array, so we could do a variety of discovery work. And that led us to interaction and an ultimate collaboration with the Regeneron Genetics Center. I mean, this started way back in 2013 when John Rico was head of SIM. Keith picked it up. Finally, we got a contract in 2019, so 6 years to get that signed. Keith gave it the name Project Generation. And this is really to do whole exome sequencing on the entire biobank, as well as a GWAS backbone. Once we got IRB approval, though, we shipped the first samples out of BAP in March 2020, and we had all the data sequencing completed in the fall of 2021, and all the data downloaded and available at Mayo starting last May. So that part went a lot faster. This includes not just the Mayo Clinic Biobank. This is a larger initiative that includes multiple PIs who had their own registries. So this is the idea, again, of how can we enhance people's registries where they can use the biobank as a control group. This included whole exome sequencing of reasonably high coverage. Again, this is of the germline. And then it was going to be a typical array backbone, but Regeneron has moved to what they're calling genotyping by sequencing. So they cover all the common SNP sites and then allow easy imputation and works as well as the mega array. So it's a very a slick way to get SNP level data. And then we're getting some additional content around pharmacogenomics genes, HLA, mitochondrial genes, ancestry informative markers, and CNVs. This is a real success. We submitted 117,000 samples and we got past QC with high quality sequencing data on, we lost about 600 participants. So this is a real kudos to BAP who did all this pulling, plating, and shipping to Regeneron for the sequencing. I'm not going to spend a lot of time just saying very high coverage. So we've got mean coverage of 48x, median coverage of 47x. 95% of the exome had greater than 20x coverage. Lots of cryptic relatedness in the biobank to be aware of for genetics people. Because the community engagement said you should return results, we are returning the ACMG 79, and we're just starting to roll that out now. And again, because we had both this mechanism to work with our CAB to get their input on how to do this, as well as our bio news, we've been able to keep people informed. And again, the return of results, of course, at the end of the day, is really up to the participants. The access to that data for the Mayo Clinic Biobank is through the standard process that I'll talk about in a moment. And then we are going to be opening this up for the entire project generation, and a good route into that will be through the SIM concierge service. All right, just a brief on two sister biobanks. So here's the Mayo Clinic Biobank, and you can see the problem. Very little diversity. So when we hit 50,000, SIM decided we would invest in some biobanks that would bring us more diversity. The first was the Sangre por Salud, or Blood for Health biobank that's in partnership with the Mountain Park Health Center in Phoenix and ASU. And that enrolled about 3,700 Latino Hispanic participants from 2013 to 18. We have bank serum plasma DNA survey and linkage to the EHR. And just to give you a few characteristics, this is a younger group and more heavily skewed towards women because they were who will commonly show up. But over 40% only speak Spanish. Very few college graduates, so lower education level, and 66% are uninsured. So a very different population than we see in the Mayo Clinic Biobank. And then a second initiative is with the University of Mississippi Medical Center called Biobank Mississippi. That's enrolled over 5,000 participants. Again, the same biospecimens are banked, and survey data and linkage to their EHR. This is a bit younger biobank as well, similar skew to ours, male to female, but 75% African American. So future directions, we are really looking to enhance the biobank. We would like to bring on additional omics around whole genome methylation, as well as metabolomics and proteomics. You know, gee, wouldn't it be nice to have that done on everybody, and then you can correlate it with the clinical and genetic data. We're planning a follow-up 3 survey to update exposures. Oops, my autocorrect here. We're going to further leverage our EHR, really expanding some of the NLP tools and starting to incorporate images. We work closely with the other major SIM initiative, the Tapestry study, which largely don't overlap, which is exciting because that really ups our numbers. We integrate with the Rochester Epidemiology Project for the Minnesota participants. So this is the population-based resource that gets clinical or medical record data from outside the clinic, from other providers. And we've cross-linked the biobank with this. So you can find out who's both in rep and in the biobank. Here's an example now of starting to annotate images. So now you could start thinking of projects that cross images with genomics with clinical data. So yeah, the project generation will be in there. At the end of the day, we are a small biobank. 50,000 is small. When you think of the UK biobank, that's over 500,000 and publicly available. We're working on ways that we can coordinate with that for investigators so that they can leverage that sort of in-house expertise. You can almost think of our biobank as either a potential pilot study to go to validate in these larger resources, or you find a really cool finding in a large biobank. Now, can you take it back and maybe do a deeper dive on the EHR or actually bring participants in? All of us is rapidly growing. You'll actually all hear about that at the next Grand Rounds with Josh Denny. And the excitement there, that's going to be about a 1 million person biobank, is there will be quite a bit of diversity in there. So same idea. However, we are looking at combining our biobank with other biobanks, particularly ones that were sequenced by the Regeneron Genetics Center. So that could bring us maybe to 500 to 800,000 sequence participants. So there's going to be these large resources as we sort of move into the next level of working in this space. Okay, finally, how do you get access to this? So for people outside the firewall, good news, all you have to do is Google Mayo Clinic Biobank and your top search hit should come up, although my Google might be a little trained to me doing this. And here you can find out, so again, I told you it's maintained for participants, but it's also maintained for researchers and people who want to propose projects. And if you want to do that, that's managed through Mayo Clinic Bioservices. So this is an arm of SIM and allows people to bring in. So for outside people, two ways: you can either come through Bioservices, or if you know somebody in your area at Mayo, reach out to them. For internal people, I also have good news. So you can actually use the internal search engine, and all you have to do is put in Biobank, and the top hit will be Mayo Clinic Biobank. And for those of you that have used this, rarely does it work that efficiently. And that'll take you right here. And here there's a lot more detail on how do I write a grant? What's the data available? Can I look at the survey? What specimens do you have? How much do you charge? And then how do I propose a project? And from here, you can submit your project. This is a messy outline. You don't really need to understand that's behind the scenes, but at a high level, the first step is feasibility. I had talked about that. We allow an initial consultation and up to 5 hours of stats time to see if you're even feasible. Now, sometimes people call and say, well, I need a Streck tube, and we're like, well, we don't have that, so the feasibility ends there. But you move along, but this really, we try to get projects in a good shape to make sure they're even feasible. Once they are, then the application is put in, and then there's an approval process through the access committee. Again, most have been approved because of that initial vetting. There's a majority rules here, but we ensure that the committee membership, voting membership, is represented. So we have to make sure somebody from the medical component or the bioethics has voted. You then need to get IRB approval for your use of those samples, and then we can send them out, and as well as send a data set. So that sort of summarizes at a high level. We've talked about the design, the governance, hopefully stimulated some ideas on how to use it. Again, it's an institutional resource and it's there to be used. And then I hope you now know how to, you can either Google outside the firewall or within the firewall to get more info. Too many people to acknowledge here, but these are a few of them. And thank you, and I'd be happy to take any questions.
Thank you very much, Dr. Cerhan. That was a tremendous amount of ground that you covered, and you did it in a very stimulating way. I think that the number of new requests for access to data, I think, is sure to go up then in the coming months. You'll be hearing from me soon, I think. But let's launch into some questions, and we have several, and I'll try to chunk some of these together. And let's start with this one. I believe that it was addressed in 2 of your slides, but as a point of clarification, could you just verify that the connections between the biobank and the rep may enable, depending on the sample studied, an integration, of the 2 resources together, sort of linking the clinical, linking the biospecimen data, and things like that into one cohort?
Correct. So Jenny St. Salver, who's one of the co-PIs of the Rochester Epidemiology Project, has come up with a way to ensure that, and they call it BioRep, projects can be efficiently handled, because there's some additional regulatory things that need to happen. But for most investigators, that'll be behind the scenes. So we've tried to make the process for developing a study as seamless as possible.
Another question addresses diversity of the existing samples, and you shared efforts at linking the Mayo Biobank with a smaller biobank with good representation from Hispanic and Latino patients. What about other groups as part 1? And then as part 2, are there plans underway for pursuing funding directly for the biobank to increase diversity and representation?
Yeah, so it's a complex question. People may ask, why were you going to University of Mississippi to get African Americans? And so clearly, a lot of our population doesn't have the diversity we need, so we need to partner. And we need to partner carefully and build a long-term trust. What we really wanted to avoid, particularly around genetic research, is this idea of helicoptering in, getting your samples and leaving, right? So we started with Mountain Park because Mayo, Arizona already had a relationship with them, and we built from that. And so that's because we really, to get the Hispanic population, particularly not a group with a different socioeconomic status. So we partnered there. When we were thinking about Jacksonville, we weren't ready yet to engage in Jacksonville because we hadn't built the deep connections we needed with the underrepresented communities. But we had a almost 20-year relationship with the University of Mississippi Medical Center that started way back with a hypertension family study and had gone on. So we built from that, and we actually had a formal memorandum of understanding before we even started this. So that was there. Looking forward, we're going to need to partner. I think there's going to be a lot of action around that, particularly as the Cancer Center is now re-engaging in our local, what we call catchments, that is the areas surrounding our home. And where do we invest? And where do we build those partnerships for long term? So I think this is going to be something that will be a really nice Cancer Center for Individualized Medicine sort of investment looking forward of, okay, here's what we have, now what do we need next? And we need more diversity, right? And so that's going to be a high priority.
Thank you. There were several questions around denominators and how the denominator is defined. I'm presuming for the Mayo Clinic Biobank specifically, and some possible limitations. 50,000 isn't a small amount, but like you say, it's not a gigantic amount either. So the first question, I think, has to do with how the denominator is defined, and it basically, I think, is the denominator geographically defined, or is it defined in some other way? For instance, there are incidence rates reported for some conditions.
Yep. So we have multiple ways we can define it, and it's really for the denominator to get incidence. You're trying to now follow everybody, so we're not geographically confined anymore. But as I sort of pointed out, if the way you're getting your incidence is through follow-up surveys, then there's a hunk of people we lost, right? If you're trying to get it through the EHR, you choose the denominator of the people that have deep EHR. Now, in the background, we are doing linkage to things like National Death Index to get mortality for people who, just stop coming to Mayo. And you can imagine that happens a lot, right? As people get frail and move into assisted living, they don't tend to come back. So we have some of that, but it's an important point, and that's why we have, if you meet with the essentially the front door, the project planning group, they'll bring those questions up, and then it comes down to, well, what question do you want to answer? Here's how we could answer it. Here might be the limitations. Does it still work well enough? Or not, it's a feasibility question. So, we have multiple sources to get the denominator, but none of them are perfect.
A follow-up question, or a question under sort of the same general idea, deals with rare diseases. Whether, sort of your opinion on whether the biobank in its current size can be used to study rare diseases.
Yeah, no, it's just not big enough. And really the way it's been used for rare diseases is people have their rare disease collection that they collect in the clinic, and they've often used the biobank as a control group. Now, there are pros and cons to that, because if your rare diseases are coming from very far away, it may not be the perfect, it's probably not going to be the perfect control group. I'll remind people there's never a perfect control group. What we always try to think about are, well, what are the biases and how strong are those biases and how much would it invalidate my result? So generally for rare diseases, its usefulness is more as the control group, not as trying to identify cases.
Thank you. If an investigator has a retrospective cohort built for a specific disease, is the investigative team able to query specific medical records based on their medical record numbers, obviously, to determine if these patients have specimens that are available in the biobank?
Yep, you'd be able to submit that project and find out if there's specimens available. You can also find out if there's genetic data. So one of the things maybe I didn't make explicit is large projects are done even by individual investigators, and they generate new data. That data needs to come back to the biobank so it can be reused in future studies. The idea being that over time, the biobank should enhance in value of the types of studies that can be done.
Thank you. And then for the diagnoses at time 0, and then diagnoses at time 0 plus 4 years, et cetera, et cetera, et cetera. Mm-hmm. How are those determined? Are they determined by diagnosis codes exclusively, some algorithm that involves diagnosis codes and other things? And I think the motivation for the question would be, for things like a hip fracture, right, that are acute, uniformly come to medical attention, diagnosis is arguably fairly straightforward. That would be one thing. But then there are more chronic disorders that are more symptom syndromes, and the quality of the codes, the completeness of the codes, et cetera, may not be like that of a fracture, for example.
Right. So what we do, so there's 2 ways to get it. One is self-report from follow-up surveys. One is from the EHR. Mm-hmm. The data I showed you, we maintain that just for those common conditions that we had reasonable codes for. They're not perfect codes, but really for your condition of interest, that really is up to the investigator. Now you'll have access to the entire EHR, so you can try your algorithm out, you can validate. There is a way to, where we call it deputize you into the biobank so that you can actually go into the EHR records. Now, what we require is once you're done extracting, you remove the identifiable information. So what we never allow a team to do is directly contact a participant. We do allow offshoot projects that would say, okay, you identified my people. We will contact them and say, hey, there's a study doing XYZ. If you're interested, now contact this new study team. Now, we still keep some control over that in that we only approve projects for specific uses. What we don't allow people to do is set up their own biobank on top of the biobank, right? Because that's going to be too confusing for the participants. But we do have mechanisms to really interact with the EHR and interact actually with the participants if it makes sense for the project you want to do.
Do you have plans to link with the CBIO portal? I hope I said that correctly.
Oh, CBIO portal.
Okay.
Not totally sure. What I will say is all the sequencing data is going to go into the omics data platform in SIM, and I can't imagine that won't be connected up with CBIO portal.
This looks like the final question, unless there are any more submissions. Are there any GWAS type studies planned on these large genomics data?
So, in the biobank itself, we've now imputed up to top meds. So every participant that was eligible for the sequencing, so some 56,000, we have GWAS data in. Really, it's project driven. People come and propose projects. We're looking at perhaps calculating a bunch of polygenic risk scores that are just widely available. But again, it's really project driven. There are a lot of groups, as you can imagine, who, when you saw that list, for example, I have lymphoma cases. I use a biobank for controls, who are doing case control discovery work for both common variants, but also rare variants from the whole exome sequencing. But yeah, it's unlimited projects you can think of. You all are creative. We've had more uses than people have come up with some great ideas that we never thought of when we launched this thing. So which is the beauty of that registry consent.
Thank you so much. By my count, I think those are all of the questions. So thank you so much for your presentation. Audience members, thank you so much for your attention and for all of these great questions. If there are no more questions, then I think that we can bring the session to a close. Dr. Cerhan, again, our deepest gratitude and appreciation for spending this time with us today.
Yeah, and thanks for inviting me.